So here's our list of terms for the life cycle of Coniferophyta. And many of these terms are actually redundant, meaning that they refer to the same thing. So a microsporangiate strobilus is the same thing as a pollen cone. Why do we need two words? Well, it's easier to say pollen cone, but we should understand that the purpose of the pollen cone is to produce the microspores. The microsporangiate strobilus. And the same with seed cones or ovulate cones. They are also megasporangiate strobili, but it's easier to say seed cone with the understanding that the seed cone is where the megaspores are produced. Okay, so a pollen cone, for example, is made up of microsporophylls or microspore-producing leaves. The microsporophylls have the microspore containers that are microsporangia, and within those we've got the microsporocytes. And whenever you see this CYTE ending, that suffix means cell in the general sense, but in plants in particular, CYTE means that's a cell that's going to undergo meiosis. And that's true here. The microsporocyte is also called the microspore mother cell because it undergoes meiosis to produce the microspore. Meanwhile, in their seed cone, we've got the megasporophylls. Within this ovulate scale complex, which is what a megasporophyll is, there are some integuments surrounding the megasporangium. There's an opening in those integuments, which is the micropyle. The megasporangium, or the nucellus, contains the megasporocyte. Again, CYTE means meiosis is going to happen here. Megasporocyte produces a megaspore. The megaspore, just like the microspore, is going to divide by mitosis to form the gametophytes, the microgametophytes and the megagametophytes. So in conifers and in other gymnosperms, the microgametophyte is reduced to the point where it is only comprised of 4 cells. So a pollen grain, a lot of people get confused and think that's the gametes, but it is not the gamete, it is the gamete-producing plant, the microgametophyte. Only 4 cells. 2 of them are prothalial cells that are associated with those air bladders that are like the ears on the Mickey Mouse face. And then in the face of the Mickey Mouse face, we've got a tube cell and a generative cell. The tube cell is going to produce the pollen tube, and the generative cell is going to divide again to form the sperm. Okay, so conifers and other gymnosperms are wind pollinated. They use the wind to get the pollen from from the microgametophyte to the megagametophyte. And in the megagametophyte, it's reduced relative to what we see in seedless vascular plants. But the megagametophyte is still big enough to have archegonia. We don't have antheridia anymore in our microgametophyte. All right, so let's take a closer look. Look at some pictures so that these words start to make some sense. So when you look at a pine tree or a redwood tree or a yew tree or a hemlock tree, what you're looking at is a mature sporophyte, a diploid organism. And in pines, the individuals are monoecious, meaning we've got one house for both the ovulate cones and the pollen cones. So within the pollen cones, we've got microsporophylls. So each one of these things here is a microsporophyll. Within the microsporophyll, we've got microsporangia. The microsporangia, remember that's the container for the microspores. How do we get from microspores? How do we get to microspores from the microsporangia? There are these special cells, the microspore mother cells, or microsporocytes, that are diploid. They undergo meiosis to form the microspores that develop into the microgametophytes, which are the pollen grains. Okay? Pollen grains released onto the wind, where they're going to meet back up with the megagametophyte, But it's going to take some time for the Megagametophyte to develop. So let's go back to the ovulate cone here. We've got the ovulate scale complexes. We've got an ovule. Within that ovule, we've got this integument from the mother sporophyte. There's a gap in here. That's the micropyle that the pollen can enter, which is what makes the seed naked, just because you have this little opening there. Within the megasporangium, which is the container for the megaspore, the megasporocyte is going to go divide by meiosis to form the megaspore. And unlike the pollen grains, where the microsporocytes can divide to form 4 pollen grains by meiosis, only one of those 4 cells from meiosis is going to go on to produce the megaspore that survives and divides to become the Megagametophyte, or the female gametophyte. So that surviving megaspore divides by mitosis to form the female gametophyte. Within the female gametophyte, we've got archegonia. This should really probably be pointing over here to the archegonium. Anyway, so the pollen will germinate, and a pollen tube will grow. And as that pollen tube grows, the female gametophyte will mature. And once the pollen tube reaches an archegonium, it will release a sperm. That nucleus of the sperm will fuse with the egg nucleus to form a zygote, our first diploid cell, through fertilization. And that new zygote will divide by mitosis to form our new sporophyte, our embryonic plant, surrounded by what's left of the megagametophyte, and that's going to develop into food reserves, or endosperm, and it's going to be surrounded by a seed coat, which is derived from this sporophyte up here. And this whole complex here is what ends up becoming the seed. So we've got our baby in a lunchbox, our embryo, our lunch, and our box. So these are the seeds that will develop into new sporophytes. All right, let's go through it one more time. We've got our mature sporophyte with both ovulate cones and pollen cones. Within the pollen cones, we've got microsporangia borne on microsporophylls. The microsporangium, we've got microsporocytes that are diploid. Those are going to divide by meiosis. Within our ovulate cone, we've got our ovulate scale complexes with the ovules inside. The microsporocytes divide by meiosis, and then by mitosis to form pollen grains. So a couple things missing in here. One is the meiosis producing the microspores, and then the microspores dividing by mitosis to form the microgametophytes, which are the pollen grains. Only 4 cells. Within the ovules, we've got a megasporangium, or a container for the megaspore, and a megasporocyte, which is diploid, and is going to undergo meiosis to form a megaspore. The megaspore is going to divide by mitosis to form the female gametophyte, or the megagametophyte, which is going to be about a couple thousand cells in size. That female gametophyte is going to produce an archegonium just like we saw in ferns and in liverworts and in mosses, which is going to be the container for the egg. The pollen, once it gets through the micropyle, to land on the surface of the megagametophyte and grow through to unite with the egg nucleus, the sperm and the egg nucleus. We get our new our zygote, our first diploid cell that divides by mitosis to form our new sporophyte, the embryo, whole baby plant inside this seed. The remains of the female gametophyte, the Megagametophyte, are going to be the food reserves, or the endosperm, inside the seed. That's all going to be surrounded by the integuments from the mother Sporophyte, which turns into the seed coat. And we've got ourselves a seed. They have these wing-like structures on here to help them be dispersed on the wind. Might find a nice place to germinate and turn into our new baby seedling. Okay, so that's it for Gymnosperms. Our last phylum is the biggest phylum. It is the Anthophyta, or the Magnoliophyta, two different names for the same phylum, in which we see the advent of flowers and fruits.